Hello everyone, my name is Alyssa Alfaro and I'm going to be walking you through 3 different examples here for Bio 207. I'm first going to start off with talking about the electron transport chain. So the electron transport chain is a chain that helps electrons move from high energy electron carriers and being transferred into the membrane proteins. So it's a sequence of redox reactions that I will be going through. So first we have the fact that this is the matrix, and then this part is considered the intermembrane space. In the matrix, we have some substrates. We have 3 NADH plus H's, which are right here. And then we also have 1 FADH2. Those are right here. Both of these substrates will be used in order to create redox reactions, and they were given from prior places within this chain. So they were taken from glycolysis as well as the Krebs cycle and the citric acid cycle, all those things. And essentially NADH becomes NAD plus and H minus, and there is an electron that is lost during that process, and that electron will be used later on. And when that happens, we have some products that are created. We have H2O as a product over here, and we also have ATP being created through a proton pump. This is also considered ATP synthesis or oxidative phosphorylation. So we have ADP plus pi becomes ATP through the proton pump. And this is probably the highest yield that will occur within the steps that it takes for aerobic respiration, because all the other ones create around maybe 2, maybe 4 ATP, but this one creates about 30 to 32 ATP, which is the highest yield. And we also have some other pumps here that I can go into depth. So we also have the cytochromes, which is this pump right here. And the cytochromes transfer about 4 H pluses, as you can see right here, per electron pair. And then we can see that there is a formation of a protein gradient used to form ATP through ATP synthase. So we have a higher ATP yield up on the top. And then we also have a higher amount of H plus due to the protein gradient. Okay, I am now going to move on to the action potentials that propagate along the axon, starting at the axon hillock. So this is the diagram that I chose for the axon hillock, which basically just shows you what an axon is. So these are the dendrites, and then it moves on to the nucleus right here, and then this whole thing is considered the soma, which is the cell body. And then this portion is considered the axon, and the axon hillock is the synthesis of the action potential, and the axon is where the actual action potential propagation takes place, and then the axon terminals, which are here at the end, are the transmission and amplification of those axon potentials. So to start off with what an action potential is, we begin at around a resting membrane potential for about negative 70 MVs. The Na and K plus channels are closed, but they're active. Depolarization then takes place to the threshold level, which causes the Na channels to open. This is a positive feedback loop for other Na channels, and the threshold is about negative 50 MVs. And MV stands for membrane potential. So at this point, this is like a positive feedback loop for other Na plus channels to open, so it just continues to get more and more and more positive until there is a plus 30 MV overshoot. And at around this point, you have the absolute refractory period, which is going on. So the channels begin to start closing and they become inactive due to the inactivation process. And then the depolarization stimulus causes voltage-gated K plus channels to open, which start to lower the MV potential. But this is considered repolarization, and the voltage-gated Na channels are now closed, but they are active. So this is considered the reflective refractory period, as hyperpolarization occurs as there is a continuation of outward movement of the K plus. This causes the potential to go below the resting membrane potential at around negative 80 MVs. Na plus K plus pumps move ions back to restore the resting conditions once again, so that they stay at negative 70 resting potentials. And at that point, another action potential can take place in order for this cycle to continue over and over again. So once one section depolarizes, the Na plus from the region will diffuse into the next region and then start an action potential there, sending it completely down the axons and being able to help the nervous system do whatever the body needs it to do. And yeah, so this is the diagram that was used and how an action potential takes place within an axon. And then now I'm going to be moving on to the cross bridge cycle in the skeletal muscle. So in skeletal muscles, you have alpha motor neurons, and these neurons send channels they send signals to the muscles in order for them to move. So essentially, acetylcholine is released by a somatic motor neuron, which binds to a nicotinic ACH receptor in the sarcolemma and causes a depolarization that stimulates a voltage-gated channels, which generate action potentials in the skeletal muscle membranes into the deep muscle fibers by transverse tubules. And the transverse tubules conduct the action potentials, which result in the opening of a voltage-gated calcium 2 plus channels, along with the calcium 2 much release channels in the reticulum, and these channels are mechanically coupled. The calcium 2 plus diffuses out of the sarcolemma in order to bind to troponin and allow for the cross-bridge formation of action myosin, which produces a power stroke. So I will be talking a little bit more about this cross-bridge formation cycle. That was kind of like the overview, but I'm going to go more into depth about cross-bridge in particular. So first we have the thin filament that's up on the top, and then we have thick filament that's down on the bottom. This is considered the myosin tail, and then the thin filament is considered actin. So essentially what we have is a power stroke is about to occur, but in order for that to happen, there needs to be some components that need to be covered. So first we have an ATP is binded in order to allow this process to occur, because it takes a lot of energy for this to occur. And also we have an actin binding site, which that is what we want to attach to the actin, but at this point it is covered by a trypomyosin, and the troponin is the thing that will make it move, but the only way that it can move is by it having the myosin binding site being able to be opened, which is when the ATP hydrolyzes, which then will go into the ADP and pi, which will expose the trypomyosin and ultimately allow the binding site to occur. So the cross bridge is considered the myosin head, having the actin binding site, is going to be attached to the myosin binding site. So that is considered the cross bridge. And when that cross bridge occurs, the power stroke can happen. And ultimately, in order for the thing to happen, the pi will be taken out, it would be hydrolyzed, and then the power stroke will happen where the ATP also shoots out at that point, and then you can see like this movement that takes place. And so that's considered the biggest conformational change, the cross bridge, and then at that point it begins to move back into the normal position that it was at before, right around here. And therefore you get a cross bridge cycle within the skeletal muscles, which allows for movement of the muscles and contractions, and ultimately the conformational change of the calcium 2+ binding to the troponin is what shifts the trypomyosin out of the way, and the myosin head gets to bind to the actin, forming a cross bridge. And the dissociation of the phosphate is the first conformational change, where the protein bends, causing a power stroke. The power stroke causes the dissociation of the ADP. Binding a new ATP causes a second conformational change. Yep. Thank you for your time.